Good evening, everybody, and this is Hearts, the exclusive channel for NEET 2021, coming with yet another super cool and super awesome lecture on plan growth and development, which is a very easy, but questions coming from this chapter are very tricky. So we have discussed in our last class regarding the introductory part of what do you mean by the growth, then the different type of meristems about the plant growth, then the definition of growth, then how can we measure growth about the different type of tissues, a small component about the anatomy of flowering plants, about which all factors can we measure growth, then in today's class, we'll be discussing all about the phases of growth. Okay, about the phases of growth. First is the meristematic phase. First is the meristematic phase. It occurs at the meristem. It occurs in the meristem of the root apex or the shoot apex. So meristematic tissue is present in both the shoot apical meristem and root apical meristem are present. So I'll show you the image for that. Okay. So in the last class itself, I have shown you several times regarding what the shoot apical meristem and the root apical meristem present in the plants. Look, this is how at the tip of the shoot, lengthening of the shoot is initiated by the shoot apical meristem, which is present at the tip of the growing shoot. And look at the tip of the root. The tip of the root also contains what? Root apical meristem, which contributes towards the growth of or lengthening of plant in the towards the soil. Is it clear? So yes, about the meristematic phase, right? So in the meristematic phase, the division or the phase of growth occurs at the meristem, which is present in the root apex and at the shoot apex. Okay. Cells in this meristematic or the meristems at the root and the shoot have a rich protoplasm. Very repeatedly asked a question. Characteristics of the cells present in the meristematic region. Repeatedly asked NEET question. Okay. So in the meristematic phase, you can find the cells which have a high protoplasm as well as large nucleus. High protoplasm and large nucleus. And you can say that the cell wall is primary. You have a primary thin cellulosic cell wall. Primary thin and cellulosic cell wall with abundant plasmodesmata. All the points are very important. So just bookmark everything. Okay, just bookmark these points. Okay, just bookmark. Is it clear? So just bookmark that particular point and study when you go further. Okay, so the cells of the meristematic tissue have rich protoplasm, then large nucleus, primary thin cellulosic and abundant plasmodesmata. Then elongation phase. Second one is the elongation phase. Okay, second one is the elongation phase. It occurs in the cells proximal, that is just next away from the tip. So elongation phase takes place in the cells that are near to the tip. That is the meristematic zone. So this type of elongation takes place in the cells which lies very close to the meristematic zone. These cells have increased vacuolation. Increased vacuolation or vacuoles are large. Okay, increased vacuolation, then size and new cell wall deposition. Is it clear? That is the importance or the characteristics of elongation phase. Last one is the maturation phase. One more will be there. Maturation phase. Okay, maturation phase occurs in the cells further away from the apex, more proximal to the phase of elongation. So maturation phase lies close to the zone of elongation, but it lies farther or it takes place in distal parts from the meristematic phase. These cells attain maximal size. These cells attain maximal size in terms of cell wall thickening and protoplasmic modifications. Okay, once again, I'll just highlight the important points for you. That is the meristematic phase. The meristematic phase, it takes place in the meristems. And these meristems are found in the root apex as well as in the shoot apex. The cells of this particular region have a rich protoplasm as well as large nuclei. The cell wall are primary and they are thin and cellulosic with abundant plasmodesmatal connection. All these encircled points are very important for your exam. Second one is elongation phase. It takes place in the cells proximal, that is away from the next, that is proximal to the tip or the meristematic zone. So these cells have increased vacuolation, then size is larger and new cell wall deposition. That is all about the next stage. Last one in this slide is the maturation phase. It takes place in the cells away from the apex. Okay. And these cells attain maximal size. These cells attain maximal size. One second. Okay. Maximal size in terms of cell wall thickening as well as protoplasmic modification. Is it clear, everyone? Yeah. One more thing. If you look at this particular diagram, you will be able to see that the detection of the zones of elongation by parallel line techniques. A and B immediately behind the apex. So suppose you take this is to be the meristematic zone. Okay, in the root tip, meristematic zone. Already told in my last class, right? My pen was not working. Okay, if suppose this portion is the meristematic phase, so this will be in meristematic region. From the region that lies close to meristematic region is zone of, one second, zone of elongation, and the rest of the parts are shown clearly in the diagram. Okay, no chance of asking this particular diagram for your exam. Okay, next, this part of the chapter is very important. Can expect 100% guaranteed MCQ from this particular slide. Okay, so it's a growth rate. So we are going to discuss about what growth rate. Okay. So what do you mean by growth rate? Growth rate is increased growth per time. It is increased growth per time. The growth rate may be arithmetical or geometrical. I have been telling, if you watch the previous videos of a channel, you might have seen that in many of the session I have just come across with this arithmetic and geometrical growth rate, and you can expect 100% direct MCQ. 100% direct MCQ from this portion. First, we are going to discuss about what arithmetic growth. In this only, following mitotic cell division, only one. So after mitotic cell division, only one daughter cell continues to divide while the other differentiates and matures. Okay, if suppose we have a particular parent cell, okay, this parent cell will divide into two. Okay, this parent cell will divide into two. Look, okay, so this cells become mature. So this cell become what? Mature, or it gets differentiated and become mature, while the other cell again divides. So the growth of this particular cell will stop. Okay, that is, it takes place and this particular cell divides into two, and this particular cell marked with the yellow, just be with them but it won't differentiate. So again, in the next stage, what is going to happen is this cell also gets differentiated, and you can see only this first cell form two different cell, whereas the latter two, that is this particular cell and this cell, gets matures. As a result, you will get only one increase, a regular increase. Okay, a regular increase. So on plotting the length of the organ against time, or the height of the plant with time, a linear curve is obtained. So what is the shape of the graph in arithmetic growth rate? A linear curve is obtained. So if you just plot a graph for arithmetic growth rate, you will obtain what? A linear growth curve. A linear graph is obtained. Look, the diagram is here itself. Okay, I just wasted my time in just drawing. Look how this is the parent. Okay, parent. So this parent plant just divides into firstly into this particular part and this particular part. So this part gets matured or differentiated and gets stop division. And at last this part divides into two. So again, two parts are formed like this. Three parts are formed. And out of which three gets matured. As a result, only a regular increase is obtained in case of arithmetic growth rate, and the graph is a what? Regular graph. And look at the case of the geometrical growth curve. The two cells will continue dividing. Suppose this is the prime parent cell. This parent cell divides into two, and this one cell divides into another one, and this into next one. And each cell, if two daughter cells are formed, that two daughter cell will undergo the successive mitotic division. Whereas in arithmetic growth rate, only one mitotic division, and after that only one daughter cell will continue to grow in the rest of the life. Okay, so we'll be discussing about the geometric growth curve in the next one. I think this was same, right? Okay, so again the growth rate is what? Increased growth per unit time, and the growth curve may be arithmetic or geometrical. So already we have discussed what if you plot a graph between time and height of the plant, you will get a graph which will, sorry, this was my problem. My pen and my hand will go in different direction. Okay. So will be a linear graph. Okay. Will be a linear graph. In mathematical form, you can express the arithmetic growth rate as LT is equal to L0 plus RT. LT is equal to L0 plus RT. Okay, this is the arithmetic growth rate equation for arithmetic growth rate. That is, LT denotes the length of the plant at time t. L0 is the time at length of the plant at time 0, where R is the growth rate or elongation per unit time. Okay. Okay. Okay, so R is a growth rate or elongation per unit time. So remember this formula for arithmetic growth rate as LT is equal to L0 plus RT. Remember the each terms, okay? LT denotes the length of the plant after time T. L0 denotes the length of the plant at a time T is equal to 0. R is the not intrinsic growth curve that you will be studying, growth rate or elongation per unit time. Very important. Okay, 100% direct MCQ comes from this chapters. Okay, next one is a plant growth, that is a geometrical growth curve. Let me have a look. 1 second, children. Sorry. Yeah, we have certain factors left out. Okay. Yes. Next, we are going to discuss about the geometrical growth curve. Geometrical growth curve means if a parent divides into two daughter cells, both the daughter cell continue to divide. So mitotic cell division takes place continuously in both the daughter cells. In most systems, the initial growth is a slow or a lag phase, then log or exponential phase, and finally it becomes a stationary phase. Okay, the graph of geometrical growth curve will grow like this: lag phase, exponential phase, and stationary phase. Okay, I think let me draw that. It's not present. 1 second. I don't know why it is not working. So followed by the lag phase, exponential phase, and what stationary phase. That's all. So here the first is a slow or the lag phase. That is, it increases rapidly and enters into log or exponential phase. And when the nutrient supply becomes limited, the growth slows down, leading to the stationary phase. This graph is a repeated times NEAT MCQ. Repeated times NEAT MCQ. Okay, 100% direct MCQ guaranteed. Okay. And what is the shape of the graph obtained during geometrical growth rate? It is sigmoid growth curve. This is also very, very important. Okay, while you are plotting the graph of an arithmetic growth curve, you will get a straight line, a linear curve. And while you are plotting a graph for geometrical growth rate, you will be getting a sigmoid curve containing three major parts: a lag phase, lag phase followed by log phase, then stationary phase. You will be asked to arrange the things, the phases in the correct sequential order. So lag phase, log phase, and stationary phase, and the shape is a sigmoid curve. So next we have the equation regarding this is a detail. So this is 100% asked question, frequently asked, 10 times MCQ, 10 times MCQ. Okay. So this is a frequently asked area from where you can expect direct questions. Okay. So in the geometric growth curve, it is a sigmoid curve. For 12th standard also, you will need the same thing in the chapter organism and population. You'll be studying about the sigmoid graph. So the graphs of biology are very important. Only 5 or 6 graphs are for you to study, and questions are frequently interchanged and asked from all the graphs. Okay. So a sigmoid curve, what is the importance of a sigmoid curve? A sigmoid curve is a characteristic of living organism growing in a natural environment. Natural environment. Okay. Sigmoid curve is a characteristic of a living world growing in a natural environment, and it is typical for all cells, tissues, organs of the plant. Okay. And what is the equation for the arithmetic growth rate? For the arithmetic growth rate, it was LT is equal to L0 plus RT. Okay. Okay. This was the growth rate for arithmetic growth rate. And for geometrical growth rate, you can have W1 is equal to W0 e raised to RT, where W1 is the final size, that is in terms of weight, height, or number. W0 is the initial size at the beginning of the period. R is the growth rate. Here it is the relative growth rate. T is the time of growth, and E is the base of natural logarithm. Okay. So this is a frequently asked area from where you can expect certain question. Okay. So let's move with the next one. So this is the growth rate continuation. So in the last, you have studied that W1 is equal to W0 e raised to RT. Here R is the relative growth rate. R is the relative growth rate. It is also the measure of the ability of the plants to produce new material, that is efficiency index. Okay. Ability of the plants to produce new plant material, or the efficiency index. And here the final weight or the final size W1, it depends on the initial size, that is directly proportional, right? So quantitative comparison between the growth can be made in two ways. First one is absolute growth rate. Second one is relative growth rate. So absolute growth rate, and the second one is relative growth rate. Absolute growth rate means the measurement and comparison of the total growth per unit time. Okay, absolute growth rate means the measurement and comparison of the total growth per unit time. A relative growth rate means it is a measurement of growth of the given system per unit time expressed on a common basis. That is example per initial parameter. Okay, initial parameters. Is it clear? Okay. So next one. So this is a diagrammatic representation of the absolute relative growth. Okay, measurement based on the given system per unit time expressed on a common basis. Both leaf A and B have increased their area by 5 centimeters square in a time to produce a leaf A dash and B dash. Is it clear? So that is the absolute growth curve or growth rate. Next is, what are the conditions for growth? First condition that we will be discussing is water. So water is required for cell enlargement, turgidity, that is osmotic balance. That is a single word. Osmotic balance. Then it provides an medium for all the enzymatic activities needed for growth. All the enzymatic activities needed for growth. Second one is the oxygen. Second one is the oxygen. Oxygen helps to release the metabolic energy for growth. Okay, helps in the process of plant by releasing metabolic energy. Third one is the nutrients. So nutrients we have studied in the chapter mineral nutrition that there are macro and micro elements needed for the synthesis of protoplasm, and it acts as a source of energy. It acts as a source of energy. Fourth one is the temperature. Okay, temperature, the plants have an optimum temperature which could be detrimental. Deviation from that particular range of temperature may cause detrimental effects to the survival of the plants. Last one is a light and gravity. Light and gravity means it affects the tropic movements at certain stages of growth. Okay, water, oxygen, nutrients, temperature, pH, light, gravity, etc. are the conditions essential for growth. So I think from this onwards, that is differentiation, de-differentiation and redifferentiation onwards, we'll discuss in our next class. So we have so many things to discuss in our chapter, okay? So much things to discuss, okay? So thank you all for your patient listening, and please don't forget to like, share and subscribe our channel, and we'll meet with the next series of the next session of this particular chapter very soon, okay?